SARS-CoV-2 is a virus we have never seen before, and the world is desperately awaiting a vaccine. However, a vaccine takes time to develop, and a short-term solution is required to slow the spread and prevent more deaths. Some researchers are turning to some tried-and-true methods to slow this pandemic, the most promising of which are antibodies. Antibodies are a special protein molecule produced by the immune system in order to neutralize pathogens such as bacteria or viruses. They are produced by B cells and are designed to be effective against only a single type of antigen through their very specific configuration in the variable region. Once a B cell encounters the antigen which matches its variable region configuration, it activates and undergoes differentiation, becoming either a plasma cell or a memory B cell. The plasma cell rapidly produces antibodies to stop the spread of the pathogen inside of the body, and the memory B cell saves some of those antibodies for future use. What we often refer to as immunity is actually how long the memory B cell lasts. But how does this relate to the COVID-19 pandemic? Well, first it's important to understand how COVID-19 works. Much like its kin in the coronavirus family, SARS-CoV-2 is a small shell riddled with spike proteins, encasing its genetic material. The spike protein docks with the ACE2 receptor of a cell, providing access for the RNA and allowing the virus to replicate itself. These spike proteins are the key to preventing the spread of COVID-19, as very specific antibodies are able to bind with them, blocking the access to a cell the virus needs to replicate. A research team led by Yan Wu at the Capital Medical University in Beijing, China, were able to isolate a pair of antibodies produced by a COVID-19 survivor. They then cloned many identical copies derived from the parent cell, referred to as monoclonal antibodies. These copies were then infused into mouse models infected with COVID-19, which resulted in a lower amount, as much as 30% less, of viral genetic material inhibiting the mouse's lungs compared to others that did not receive the same treatment. Of the 4 unique antibodies that were identified, B38 and H4 were seen to be able to bind to vital parts of the spike protein. In theory, these antibodies can be donated by COVID-19 survivors in forms of plasma, which can be used to possibly prevent or help those suffering from the virus.
Donating plasma, however, holds its own set of risks. The transmission of bloodborne diseases such as syphilis, hepatitis, malaria, and many others have been a cited problem with this method of treatment. However, modern blood banking practices provide the proper security needed to prevent the transmission of these diseases.
During this time of uncertainty, it has become increasingly hard to see the light at the end of the tunnel. However, always remember that there are people working tirelessly all over the world to keep you and your family safe, whether that be in the form of antibody treatments, vaccines, or social regulation. We are all in this together.
Thank you.